Hello, is it on? Okay. Sorry. Hi, everyone. Good afternoon. Welcome. Thank you for joining us today on this lovely sunny day in California. No, but really, thank you for joining us today. I know it's all a bit of a trek for everyone, especially with the weather. So, for those who traveled near and far, thank you for joining us. So, welcome. I'd like to acknowledge our special guest today. We have a councilman from West Covina, Councilman Ollie Cantos is joining us here today. And we have a delegation from the STEM class of Mayfield Senior School who's joining us here as well today, along with their teachers and Assistant Dean of School. And I wanted to welcome my biology teacher, Miss Teresa Peters, as well for joining us today. Um, so today's symposium is also co-hosted by Dr. Emily Wiley, fellow Fulbright alum, and of course, professor of CMC for the Kravis Day Integrated Science Department of CMC. But before we begin, I wanted to invite Dr. Wiley to say a few words.
Hello, everyone. I just had to welcome you all. This is a really big moment for us. It's one of the biggest events we've hosted in our new building. I'm the Director for Program Development for the Kravis Department of Integrated Sciences here at CMC. And we welcome you to explore the building afterwards. There's 3 floors above this one. And it's built to inspire kind of the main mission of this program, which is work at the intersections of the natural sciences, the social sciences, and humanities, and also work on complex problems that require a lot of interdisciplinary thinking. So I hope you'll appreciate all the intersections you see as you explore the floors. Anyway, it's wonderful to see you. I'm very happy that we can share this new building. It was opened 3 months ago. And it's a big moment for us. Thank you for being here and coming out in the rain.
Thank you, Dr. Wiley. So today, as we celebrate International Education Week, we're reminded that when people learn from one another across borders, the world changes. So the Fulbright Association has championed this truth for decades, showing that global exchange isn't just about education, it's about unlocking human potential. So today we're going to highlight a few of these values via full scientific discovery. Few have embodied this better than Dr. Francis Collins in this photo above here. So this was during the Fulbright Prize ceremony that was held in Washington, D.C. So Dr. Collins is actually one of the prime leaders of the Human Genomics Project. And at the NIH, he helped to spark the era of precision medicine. Therefore, our event for the symposium today. So today we highlight how these values fuel scientific discovery, and the symposium will bring these threads together for international education, the Fulbright spirit, and the scientific breakthroughs that emerge when knowledge moves freely. So I'd like to welcome our panelists today. So we have Dr. Barbara Fortini, Dr. Patrick Fueger, and Kurt Jose. So Dr. Fortini is the program director at the Master of Science in Human Genetics and Genomic Data Analysis program at Keck Graduate Institute. She's also a former professor at the Keck Science Department, and Dr. Fortini's research focuses on non-coding genomic variation effects on colorectal cancer risk. Dr. Patrick Fueger of City of Hope is Associate Professor in the Department of Molecular and Cellular Endocrinology in the Arthur Riggs Diabetes and Metabolism Research Institute. He's also the Associate Director of the Cancer Metabolism Training Program. Dr. Fueger's research and his team of researchers at the Fueger Lab are hunting for therapeutic targets to prevent or reverse a series of metabolic diseases such as type 1 and 2 diabetes and fatty liver disease, all of which lead to deadly forms of liver cancer. And we have also Kurt Jose, the founder and CEO of IndiviDx, a biotech-driven clinical laboratory specializing in precision medicine. He has dedicated over a decade to bridging the gap between science and everyday care. So at IndiviDx, he has seamlessly integrated biotech innovation with clinical expertise to promote personalized and human-centered care. So I'd like to welcome all our panelists today. I'd like to welcome Dr. Fortini.
Yeah. All right. Well, thank you everyone for coming today, and definitely thank you for the invitation to speak to the group. I really loved the title of this panel, so I stole it as just my first slide because I do truly believe that not just the future is personal, but the current state of affairs should be personal. So I study colorectal cancer, and one might ask why to study colorectal cancer. Well, it is a very important public health issue in the United States and in the world. It is the third most common diagnosed cancer in the United States. It's also one of the top causes of cancer death. Of course, I study the genetics of colon cancer, and colon cancer is the most environmentally influenced of all the cancers because it matters what we eat and what touches our gut. But colon cancer has been in the news a lot recently, especially as it relates to younger people. And so, of all cancer deaths in people under the age of 50, colorectal cancer is now number one in men and number two in women. And there's a lot of research going on right now and debate trying to understand why that is the case, because in most of our cancer types, we have decreasing rates going on in the United States. And this is a difference, something that's climbing. So I use genetics to try to understand how colon cancer develops, and we look for in our lab the missing heritability. So these are the types of reasons you might have colon cancer that we don't know about. So most colon cancer is what we would consider sporadic. It is not caused by an underlying genetic risk. It may have to do with random chance. It may have to do with whether you don't eat enough fiber, whether you eat too much delicious processed meats, which is a risk factor for colorectal cancer. And we also know that there are some highly penetrant cancer syndromes. So these are genetic inherited variants that might cause you to have an extremely high rate of colon cancer. And those are our syndromes called FAP, or familial adenomatosis polyposis, and there are some others like Lynch syndrome. But even though for the people who have those variants, they're extremely highly at risk, it does not explain why most people get colon cancer. That's still about only 5% of the cases. And so through doing twin studies and looking at how often cancer is heritable, we know that there are some genetic risk factors that we don't know about that are in the population. And so those are the cases that are circled with familial risk. And so for those of you who've taken genetics, and if you learned about narrow sense heritability, different types of studies in different populations put that genetic risk between 12 and 35%. So how do we find those risk factors? Well, in the early 2000s, we started doing things called genome-wide association studies, or GWASs. And with the GWAS, instead of studying small families that have lots and lots of colon cancer, we started looking in giant populations to see if we could use sequencing to identify markers that are more common in people that get colon cancer than in the population that doesn't get colon cancer. And so some of the studies at the beginning were very small, but by the end, the last one I was involved with, we had over 100,000 cases and 180,000 controls. So that's the size of the experiment. And we're looking at genetic markers that are spread throughout the whole chromosomes, our whole genome. So we would look at hundreds of thousands of loci. So looking at the effect, most of these were single nucleotide polymorphisms. And so we had some that were general and some that were custom that we kind of knew about some cancer genes. So we put a lot of markers near those genes just to really cover them well. And so by doing this, we found there are, in fact, over 100 places in our genome where if you have variants, you might be at a slightly higher risk of colon cancer. So in my research lab, we try to figure out why. Why is this one variant that's in a region between genes going to give you a little bit higher risk of colon cancer than the average person? And so this type of study has led us to new types of biology because it was a statistical test. We did kind of seed the field a little bit by putting markers near our known cancer genes, but again, we looked throughout the whole genome without having a hypothesis for every place. And the places I was really interested in were the surprises. You know, we have variants near famous cancer genes like MYC or some of the SMAD family, the TGF-beta. That told us that the technology works. We find things involved in cancer, but then we found variants in places where we had completely unknown, undescribed genes. We also have some in genome deserts where there's not a gene for 1 million base pairs in either direction. Haven't figured that one out yet. Still going to work on that. But we use all kinds of different genomic data sets to help us discover these novel genes that are impacting colorectal cancer risk. And so we look to see other variants in the neighborhood that are part of the linkage disequilibrium group. Those are just the friends that are always inherited together. We can use chromatin epigenomics to tell us which part of the genome might be active in colon cells. And then we can look at gene expression data to see if people with the risk variant maybe express too much or too little of the genes nearby. And by doing this, we've actually discovered a few novel genes. And so my lab works on trying to understand what those genes do. But that's not really precision medicine. So I don't do a lot of precision medicine in my lab. But this same data can help people because we now know over these 100 sites, we can genotype individuals and say, did you get super lucky? And did you get all of the low risk versions of those 100 variants? Or did you get super unlucky and you got all the high risk versions? So it's not so much that you get one or two, but if you look at over the entire population, we get a little bit of a bell curve, a little normal distribution. Most people have some of the good variants and some of the risk variants, but there are those small tails where we have some people that got very unlucky. And so when we have that information, we can actually tell people, you know, you might be in the top 20% risk group. So perhaps you should have your colonoscopy a little bit earlier than maybe other people. We can also start to put together lifestyle data with this. And this is something that in the colon cancer field, we're not doing quite as much as other fields are. So if you were getting some testing for like cardiac disease, they would maybe help you understand how your variants interact with your clinical data. Maybe if you were being screened for breast cancer, there's a tool called Boadicea that would put a bunch of data together. But here we do know that there are lifestyle risk factors for colon cancer, like not eating enough fiber, heavy alcohol use, or if you have an inflammatory bowel disease. And then we can add in our percentiles for did you get the low risk version of the polygenic risk score, or do you have the high risk version? And then we can add those together. So if you had a very high polygenic risk score, you might choose to either have different types of screening, so maybe get your colonoscopy more frequently or earlier, or you might decide like, okay, I am at a higher genetic risk, I'm going to make a lifestyle change, like making sure I definitely get enough fiber so I can make my environmental contribution as safe as possible. So this is sort of the closest we get to precision medicine right now in the risk prediction world of colorectal cancer. But because this is talking about just how we use genetics and genomics in general, I wanted to be precise about what I mean by precision medicine. I think it's one of those terms that everybody has their own definition of what counts as precision medicine. So what I'm going to use as my baseline definition is that you're going to recognize our individual variability of genes when we're thinking about all of our health risks and how we're going to respond to them and how we're going to choose treatments. And we can use those differences to predict our individual risk and maybe in the future even prevent incidents by using that data. If you happen to be diagnosed, maybe you're going to use now a treatment that's customized to you. So that customization might be still shopping from a a shelf, right? We're not going to necessarily make a treatment just for each individual cancer patient, but in precision oncology, we might look at your germline variants, the ones in all of your cells, and then the ones in your tumor, and decide, okay, this is the top line treatment we're going to try for you based on those variants. We could also use pharmacogenomics to determine which dose of medication we're going to give to people. We know there are genes that help process all the drugs that we take, and some people process them faster and some people process them slower. And so there could be an individual dosage even within the same medication. And then for people who do have rare genetic conditions with a single genetic cause, and we could put that gene back, we do have a few gene therapies now that are a way to cure or fix the problem with the genome. So the real question is, will this improve outcomes? I think everyone in this room hopes that that is the case. But how will it? Because we want to make sure that we're not just doing this to feel like we're using science, but we need to actually see that there is higher efficacy by matching our treatments to the molecular mechanisms that are driving someone's disease. We also want to reduce adverse outcomes. Going back to the pharmacogenomics, if you're going to process something more slowly, you might have a higher sensitivity to that drug. So maybe we choose a different one for you. And then we have this dream that things will eventually be lower cost because we're going to start with the optimal treatment first. We're not going to try a bunch of different things till we land on the right treatment. And so by doing the optimum thing as our first line of treatment, hopefully we'll reduce how long people need to stay in hospitals, reduce how long people need to be on a treatment, and that will lead to improvement all around. So the question, I kind of led with this, are we in the age of precision medicine now? Well, we finally have the $100 genome. So this record was broken a couple of years ago by Element Biosciences. There are now several different companies that at scale, so if you're doing at least 1,000 a week, you can deliver them for $100. There was now, we don't have an exact number of how many people in the world have had whole genome sequencing, but the estimate that I got off of ChatGPT was that it's over 2 million. Especially if you consider we assume that there's lots of sequencing going on in Asian countries. We know how many people have been put into the UK Biobank. We know how many people are in the All of Us database in the United States. And so we have a lot of data now that we can start mining for information on the genome and phenotypes and outcomes for people. Sequencing is also very fast now. There is a new record for the time it takes from when you start sequencing to when you have a completely interpreted report, and that is now 4 hours. And this was done at the Broad. Stanford had set the record a few years ago with 6 hours. But because some of our new technologies allow the data analysis to start on the sequencer, we don't even have to wait for sequencing to finish and then copy the files over to another computer. All of that's integrated now. We can have results in a clinical cycle, right? So you could come to the hospital in the morning, you could have a problem, you could be sequenced by the time your doctor rounds at the end of the day, right? And that's going to change things. We also have other types of data that collect information on our health. How many of you are wearing a smartwatch that tracks your steps and your heart rate? Mine can even do blood, oxygenation, right? So we have other types of data that could be involved in making healthcare decisions that we now can just integrate into our lives without having to have a separate visit to the doctor to analyze how our treatment is going. Thinking about the rare disease therapeutics, there are now 13 FDA-approved gene therapies. These are single gene replacement therapies. If you add cell therapies and some other types of genetic-based approaches, we have more than 40. And of course, the big one is AI. How can we automate some of this that we don't necessarily need to rely on a single grad student sitting in a basement going through files trying to figure out what's going on? We can have some of, as I said, the sequencer is already starting the analysis as it's doing the sequence, and so things are interpreted by the time we collect the data, which is very exciting. What are the barriers to actually doing it? Well, it's still the same barriers that we've always had, right? Social determinants of health, access to these kind of centers. These tools while they are steps in the right direction, they still haven't made this the standard of care for every patient who walks into, especially like a small clinic that is not part of a major research center. But I like the fact that there's lots of young people because you can go out and fix that. You're going to change it, right? So I wanted to talk a little bit about how to get into the field and some of the aspects that maybe people don't think about. In order for us to make precision medicine standard, we will need doctors. So if you wanted to become a geneticist or a molecular pathologist, that is a great way to do precision medicine. You'll have to go to med school and you have to pass your boards and do your residencies and fellowships. But even if you don't want to go through those steps, but you want to be involved in clinical research, there's lots of clinical research trials that are being run by nurses, nurse practitioners, people with master's degrees, or PhDs that are looking at how we do clinical delivery of care and actually asking, was there better outcomes? We also have a great career path of being a genetic counselor. So one of the hats I wear at KGI, we have a master's in genetic counseling. I'm involved with that program, and these are the professionals that help individuals understand their genetic information and make informed decisions that uses that information. So if you wanted to be like a navigator that would help people go through that testing process and know what to do and really connect with the individuals, there's genetic counseling. If you really like the detective work and you want to know, does this change in the DNA actually lead to this disease? There's a whole career called a variant scientist. If you want that job, you can either have a PhD or you can get a master's. Come talk to me about our program in genomic data analytics, or we have some future variant scientists sitting over there in the corner. You could talk to them about the program. If you really want to understand how the genome works, and so we have the basis to form these new therapies and treatments, of course, being a researcher, that's what I do every day. I have a PhD. But if you want to be more on the therapeutic discovery side, you could work at a biotech company, and there they have roles from master's to PhD. And then a big part of how we deliver genomics-driven care requires bioinformatics and software. And so if you're really interested in software development for healthcare, there's a huge need for people to make all of the interfaces, all of the pipelines, all of these protocols that will put the AI systems into place so it can help patients. And then I'll give a shout out to the other side. For a lot of these gene therapies and these molecular based medicines, you have to, they're very expensive to make. And so there is a whole field of bioprocess engineering that's involved in the synthesis and the safe production of these kind of high-end therapies that are being delivered to patients. And so that would be more of an engineering pathway, but again, in the biosciences. And so with that, I am very excited to hear from the rest of our panelists and happy to take your questions later.
Thank you, Dr. Thank you, Dr. Fortini. I'd like to invite Keck Jose of IndiviDx.
All right. Good afternoon, everybody. I want to first thank the Fulbright Association, Keck Graduate Institute for having us here today on this panel. And so today I'll be talking further about our work at IndiviDx and what we do, I guess, in the private and commercial sector. So with that being said, let's begin. So first I want you to take a step back and imagine a world where your DNA is the key to safer, more effective care. And within that key are answers that help you prevent harmful reactions and guide truly personalized treatment. Now, is this something we have to imagine? No. The truth is, we don't have to imagine because it's a reality. That's why this panel's here today, and that's why we're all having this discussion today, because precision medicine is here. It unlocks this potential by using the keys in your genetics to become a powerful tool that now transforms healthcare from guesswork into hope, clarity, and life-saving precision. So now I'm going to be talking to you about what I titled One Size Fits None, when DNA becomes part of the solution. And with that today, I'm going to be talking about the missing piece, which is our DNA. It becomes the key that we've seen at IndiviDx to where we move from beyond one size fits all this era of precision medicine, and I'm going to further unpack what pharmacogenetics is, or PGX for short. And then I'm also going to discuss trial and error to targeted care, the implications of this data-driven decision making, and how has it changed the landscape of the healthcare field, at least from the clinical lab perspective. Then I'm going to go on to the current barriers in pharmacogenetics that we've seen over the past decade. Some of those barriers include regulation, education, and implementation. And I'll also share like what we have seen firsthand, successes, pros and cons. And finally, I'm going to close with building a healthier tomorrow, a promise that's always there, but now more than ever, we're closer to delivering it. And I'm going to be discussing what's next in the field of precision medicine, AI, pharmacogenetics, and how all of this will come together. So first, we're going to start with beyond one size fits all. So I'm going to give a little bit of a history lesson because, just to clarify, I'm not a doctor. I'm actually a lawyer. So so from the 1950s, they first started to see varied responses in tuberculosis drugs, and then in 1961, Werner Kahlo. You first started to hear the term pharmacogenetics. He started to see how these genes and drug responses were so varied in people. Now, fast forward to the 70s, 80s, 90s, we now started to see the development of CYP450. That's the liver enzyme, that's basically the machine in all of our bodies that metabolize these medications. And it's rooted in our DNA. So now if you think with me for a second, that's the factory in there that's processing all these medications. And finally, in 2003, we have the Human Genome Project. All the genes successfully mapped in the human body. And we owe that work to Francis Collins and everyone in the UK and the US to drive that. And then with the Human Genome Project, that really accelerated, as you heard earlier, how that changed the game. And finally, in 2015, President Obama was the sitting president at the time that launched the Precision Medicine Initiative. That was a major shift from one size fits all. And it was, which is the status quo, to a more tailored and personalized approach to healthcare. It revolutionized how we approach healthcare, how we approach disease, how we approach research. Now, through that initiative since 2015, it bolstered efforts to accelerate technology development, and it also led to companies like us, where we're now able to provide this technology and molecular testing as part of patient care and make it readily available to the public. So it bolstered all of us here on this panel to accelerate and move forward. But also at the same time with this question, it brought new questions of genetic data privacy, implications of collection and storage, which are still questions today. So this is just a slide talking about the cost of genome sequencing, because it did greatly reduce from 2003, whether it's what we do in pharmacogenetics or other tests we do in PCR syndromic, where we identify certain viruses. It really allowed from one size fits all to predictive, preventative, personalized, and participative diagnoses. So to unpack pharmacogenetics a bit, if we just imagine here this one pill, people have varied responses. They go from normal, some respond to a lower dose, some a higher dose, some need an alternative. But throughout the decades, we've always been based on population. We're based on age, weight, height, and whatever they've done during trials. But now they're trying to refine that and adjust to the actual unique responses we have. So now that takes me to IndiviDx. That's the company I founded. We're a biotechnology-driven lab that specializes in precision medicine, more specifically, diagnostic tests such as pharmacogenetics, PCR syndromic, and other types of molecular tests used for diagnostic purposes. One thing I do want to point out, we are clinically certified. We're not like what you see direct to consumer, like 23andMe or Ancestry. The work we do is medically actionable, and our goal is to really foster effective treatments for clinicians, for practitioners, and help improve human health. And for the past decade, that's precisely what we've been doing, is trying to improve the medical community through those personalized approaches in a timely manner. So this is just some of our certification. We're a biosafety level 3. We're headquartered in, well, I have Southern California, but we're only 10 miles away. We're not too far. And these are just some of our qualifications, and we really focus on precision, dependability, and innovation. So our main mission really is deliver evidence-based treatments to the medical community we serve and raise awareness. We want people to be informed, we want people to be part of the dialogue, and we also want them to understand how this impacts them, because at the end of the day, most of this is powered by all of you and all of us. It's not static, it's dynamic. So now, I know you heard the definition of pharmacogenetics, but I'll unpack it a bit more since it's our specialty. Essentially, pharmacogenetics testing, at least for us, is a cheek swab. And it's as simple as brushing your teeth. In 30 seconds, you swab each side. And through that cheek swab, we examine drug concentration, efficacy, and toxicity. So we kind of see how you metabolize the medications, how you absorb it, how you excrete it. Through efficacy, we see what's effective, what's not effective. And then through toxicity, we can see what levels are toxic for you or what might not be toxic for you. So that's what pharmacogenetics does. Now, it's not a panacea, but it does help with the following issues. So with these multitude of issues you see on the board, adverse drug reaction, trial and error prescribing, high costs of healthcare, polypharmacy complexities, gene-drug interactions, and underutilized personal care. Now, each of this could be a symposium panel, but I'll try and unpack each one. So adverse drug reaction is the fourth leading cause of death in the US, 4 to 6, depending on what's going on. Unfortunately, causes anywhere from 100 to 125,000 deaths annually. Now, when it comes to trial and error prescribing, you see that more so when it comes to mental health, pain, cardiac. It's a lot of issues where there's questions of medication support, questions of compliance, questions of what's effective, what's not effective, and then high costs of healthcare. Now, if you think about all those effects in trial and error prescribing, now you couple them with repeated doctor's visits, you couple that with stress on family and friends, you couple that with medical debt and policy, and then now you look at polypharmacy issues, which is more prevalent in people 65 and over. That's where pharmacogenetics aims to address these issues, because it can kind of see this before it happens. And that's what we've seen firsthand. We've seen that with, which I'm going to go on later, on how we've seen that, like where it's made the most impact. And for some people who had pharmacotherapy failure, we've seen evidence where patients who don't respond correctly, they can now have a substitution medication or remove that completely. And then when gene-drug interactions, we see how patients with those specific genes have specific errors. So basically, the doctor can be informed on how to make adjustments. Sorry, oops, underutilization. So to start with trial and error, basically this is kind of a preview. It's a cartoon, but it actually highlights what we do. So that cheek swab, when it comes to pharmacogenetics, or PGX for short, it has value throughout your life because your genes do not change. For the most part, if anything, you'll be doing the test later because the technology changed. And that's what we've seen over the past 10 years. But through each profile, we do this in-house. We take the DNA, we sequence it, we extract it. Extract, sequence, then we amplify it. Then from there we see, we come up with this report. This report has risk scores. This report has certain implications, which we then now give to the doctor or the clinician who ordered it, whether it's a nurse practitioner, a doctor, whoever it may be, pharmacist. They now have this information they can look at after we've done unpacking the bioinformatics of it. So with that, it provides insight, whether you're a non-responder or you respond well, or you have a chronic condition. But I should highlight, even if you do not have a pre-existing condition, it also is useful as a form of future proofing, or what we hope would happen later on, where it does change the standards of care. So when you come in to the emergency room, you now have a new baseline. You're not just an unknown person. They actually know more about you than what happens today. And also, pharmacogenetics is cost efficient. We've seen that where people have been firsthand where they're not spending on repeated visits. So in some instances, for the elderly population, they're even able to deprescribe and lessen their medications. So with this ecosystem of healthcare, there's over 1,200 FDA-approved medications with genetic markers. So that's where pharmacogenetics, FDA, and medications really work hand in hand. And let's see. So this is just some studies I just wanted to share with you, and I could share these sources as well. But they did one where there's antidepressant treatments for patients who receive PGX, and the results essentially were they saw 41 to 78% more likely to achieve remission, 20 to 49 more likely to respond. And then in another study, they saw fewer ER visits, but they actually saw a boost in outpatient visits. Now you're probably wondering, wait, why? I thought he just said it's cost effective. Why is there more outpatient visits? There's more outpatient visits because now there's guided treatment in between. So even if there is more visits, the doctor's there, the genetic counselor's there, and they're helping you guide your treatment. So yes, you may have another visit, but think about it. If you're not having a headache, if you're not having dry mouth, or if you're not in the hospital for possible kidney failure, it's worth it to have that extra visit, even if doctors' offices are not the most fun sometimes. So this is actually a real person in our profile that's been anonymized, but I just wanted to show you a bit about what we do. So this is just part of our report where we categorize standard precaution, precautionary use, and high caution. Now this individual here is actually a real case study, and we found out that they had a very adverse drug reaction to warfarin, which was interesting because that comprised of most of their treatment plan, and it triggered adult depression. It also led to 10 plus years of suffering for this person. And the team, we were talking like this a few years ago, and someone walked up and they said, hey, can my family member get this test? And we said, sure. And when we found this information out, we saw that there was 3 meds that were really causing the problems, triggering these mental health issues. So rather than fixing the pain, which actually originated from pain management medications, not even psych meds. So when we saw that, we said, oh, okay, we can make adjustments. So we made some adjustments, and within a matter of, well, actually, you might have heard earlier, it really does take anywhere from 4 6 hours, less than a day. We found all those insights and we gave it to their clinician. After we passed on that information, the doctor started adjusting, removing certain regimen, adding substitutions that they thought of, and within a month, they started to see that now there was no episodes. Within 3 months, now we saw no more psych meds. Within a year, no more pain meds, and they started to respond better to their rehab. I should also emphasize this person had a very long-term injury caused from work and required multiple surgeries. So with that being said, after a year, they were not on any medications. The only memory they had of this whole ordeal was a cane, which nonetheless, even though that's not the most ideal scenario, they were able to live their life. He was no longer in isolation, no longer having episodes, and they could be part of their family again. Now, as you see here on this chart, you'll see genotypes, phenotypes, and that's how we kind of assess how people metabolize medications. So like I said earlier, CYP450 is the core, but within CYP450, you have CYP2D6, CYP2C19, CYP1A2, and I know I'm throwing out a lot of codes, but CYP2D6 metabolizes 65% of most of the prescribed meds you would take in your lifetime. And like for instance, 2C19, which you might see up here on the top, this person is an intermediate metabolizer. A lot of that enzyme is relevant for like Lexapro and Zoloft. So if you know anyone who's taken any of those, it's pertinent to it. And being an intermediate metabolizer means that they metabolize it more than the standard dose. VCOR C1 for this individual, for instance, warfarin dosing difficulties is what came out. So out of all the medications approved by the DNA, roughly 1 in 5 have genetic biomarker information. So one thing that has changed in, when I say how technology changed, this is actually something new that we've added. We now, for the past 2 years now, we've been able to incorporate lab data, lifestyle data. So we can now put kidney function scores, for instance. We can also put different types of medications, and we can also go by condition. So, for instance, let's say you did a test today and you had no issues whatsoever, but a year from now you had a broken ankle or you went to the dentist and they gave you medications, we can put that in. Or let's say you were given something to boost mood or to help with depression. We can now put those specific conditions in, isolate it, and tackle it so that when your doctor does call us, whether it's your GP, whether it's anyone, we could now help them and guide them. And when it comes to cost, we could also look at the particular cost of each med based on the database. So that's where AI is really going to make an impact in terms of trends and finding patterns. But as you can see here, we have all the different things, and in red is excluded therapies because of one of the issues with this particular individual's genes. So now this is what we call healthcare powered by you. It's genetics, it's actionable information, it's clinicians, it's a treatment plan, but it's all powered by you. And that's what we've seen in our experience, how it's helped with effective decision making. And now I'm going to talk about current barriers in pharmacogenetics, or PGX. So that's the knowledge gap, the underutilization in practice, provider training, tech issues, data privacy. So this knowledge gap has always existed. It still exists today. I mean, it also shows in the stats, like for instance, in Medicaid and Medicare, PGX has been used 4% of the time in Medicaid and 10.5% in Medicare. So it's still very underutilized even today. And the next question in this knowledge gap is, well, we have the result. What do we do with it now? And that's been one of the issues that even we've been tackling from day one. Yes, we provide the result. Yes, we work with doctors, but then at the same time, we don't have, we would need to train their staff. We would need to speak with genetic counselors. If they don't have that there, we kind of have to build it from the ground up for them. Or we also provide the service constantly throughout this whole process. So, and then in regards to IT issues, there's no seamless integration across health systems. We have a report, but it's not within the whole health ecosystem of, let's say, a hospital. It comes in a PDF format. It's not like you can seamlessly integrate it into any EHR at any hospital or any research institute. It's you could, but it's a very hard workaround. And then lastly, genetic data privacy concerns. With the rise of more direct-to-consumer testing, it led to questions of who holds this data, what can they do with it? And I'm sure all of you have heard of like 23andMe, where they've sold people's data without their authorization. So what are the solutions? This is what we've seen firsthand. So bridging the knowledge gap, it comes to situations like today where you're all here participating, and we're all grateful for that. It comes to engaging with doctors. It comes with more on-the-job training, CE credits, degrees, whatever it may be, to help bridge it. And underutilization, that just comes down with speaking with policymakers, engaging with organizations to help provide more standardization and more studies. So for those aspiring researchers here, more studies will show how effective it can be. And for those who want to go in the commercial field, it's also needed because we can demonstrate how effective this work is to people. Now, in regards to tech, I've had to tackle this personally being a cybersecurity specialist. We've had to create our own internal protocols, have a chain of trust with verifiable entities and encryption. But most, like for us, for instance, we've helped guide some public agencies on how to handle genetic data. We're actually one of the first companies that started the whole notion of deleting your data after testing. And even till today, we maintain that. So once your testing is done, that de-identified data, we will delete it, even though we have an obligation to hold on to it for 5 years. But we have, we will we'll remove it from our system, and we also try not to keep everything on the cloud. We keep it in physical servers so that it's not easily accessible. Now, with that being said, what's the future for precision medicine for all of this? And the future is that close as we reach closer to that promise of a healthier tomorrow, where we want to standardize PGX testing for more targeted things. We want to have medicine and treatments that become more personal, and as we're going to see more, that's where we're going from the research sector to here, and improvement in implementation. It would be great to see a world where PGX becomes point of care for your records and becomes part of that baseline, um, which then leads to more targeted therapies. And and in the future, we hope that with more developments, we'll be able to take more lifestyle, more genomics, more microbiome, more epigenetics. We can now put it in. And we hope that legislation does that as well. And with the development of AI, we can uncover more patterns for value based care. So, this is just an infographic from the AMA about precision medicine. And then these are just some articles recently of what's going on. Like, for instance, from the VA, that was yesterday, where the VA now is starting to use it. For instance, this individual, U.S. Army veteran Darrington Robinson, he had high cholesterol and wasn't responding well to his medications. And Dr. Newberry from the VA worked with him, and they were able to come up with more effective therapies and provide counseling, which to me was actually new because the VA didn't really have that a few years ago. So it's nice to see. And then in the NHS in England, they're talking about implementing pharmacogenetics for pediatrics. So now, how do we build a healthier tomorrow? Which I'm going to close on. Uh, as you've heard earlier, you can be part of contributing to research, to the All of Us project, other research institutes. You can participate like today, where you're seated, because education is the key. And then at the same time, it's starting the dialogue. At IndiviDx and for myself, we want you to be part of the dialogue because this data, this testing is all powered really by you. But that's the best part of precision medicine. It's science powered by you. So you should be part of the dialogue. And I think that's the biggest shift in healthcare, at least from my perspective. And at the end of the day, as long as we're all part of the dialogue, we can find those answers. So, yeah, so I want to thank all of you. Appreciate it.
Thank you, Mr. Jose. I'd like to invite Dr. Patrick Fueger.
Okay, we have a minute or two, so I'm going to try to truncate some things and talk about a hope for precision medicine and diabetes. Where cancer is today, I think we're at least a light year behind in diabetes, but hopefully we can catch up in the next few years. So I have nothing to disclose other than in the first speaker's presentation, I'm the rodent in the slide. So I'm a drug hunter. I'm a I'm a molecular physiologist by training, but I work in the preclinical space. So things with mice and rats, and I'll hopefully most people know something about diabetes. Part of our big problem in where we are and how far behind we are in precision medicine is we've really have struggled at genetically diagnosing these diseases. So the two main players are really the main player in both forms of the major forms of diabetes, type 1 and type 2 diabetes, are the pancreatic beta cells. And I'm not in my reading glasses today, so I may get a couple subtle things wrong, but it's the cells in blue. In the middle of this little circle here, this is the islets of Langerhans. So they're little microorgans within the pancreas. And in the case of type 1 diabetes, these cells are killed by an autoimmune attack. In the case of type 2 diabetes, it's a little more complicated, but ultimately the beta cells fail, just like in type 1 diabetes. So insulin comes from the beta cells. That's the major hormone that lowers blood glucose, so moves it from the blood into tissues. And then glucagon in the alpha cells, which I think are in green, is the converse hormone that leads to glucose mobilization. So those two are kind of yin and yang and balance against each other. So part of the reason we're way behind, as I alluded to, is that we really had type 1 diabetes, type 2 diabetes, and now there's some monogenic forms of diabetes that if you're actually diagnosed with something that looks like type 1 diabetes before 6 months of age, at least in the UK, your DNA gets sent off and they look for a single gene defect that you have because you don't really have an immune system until you're 3 to 6 months old. So we used to think everyone with type 1 diabetes was the same. We thought it had a very rapid onset, so maybe month or two, and that led to the disease. Type 2 diabetes on the bottom is a little more complicated. You first have what's called insulin resistance, where the insulin from the beta cells is not as effective, and so you have a compensation period where you start making more, you make more beta cells. Eventually, that fails, and you go below this dotted imaginary line, and that's when you develop type 2 diabetes. So, type 1 diabetes we thought was very rapid. It turns out that's actually wrong. And it depends on so there's very strong heterogeneity of the disease. So children who are diagnosed with type 1 diabetes have a more acute drop in beta cell failure. Whereas if you're diagnosed with type 1 diabetes later in age, the slope of that line is actually a lot more gradual. So at any rate, there's many other things that we could have a whole symposium on just the different subclasses of these diseases and how we're getting closer and closer to actually be able to genetically characterize these diseases. So again, we used to think it was a very steep fall in beta cell mass. We now have different stages of the disease. Well, it's called phases here, but there's stages today. Stage 1, stage 2, stage 3. And it's important to actually know where the patient lies into those stages. One thing we also struggle in is being able to predict who's going to get type 1 diabetes in this case. And so can we identify those patients earlier? We're certainly on the way, and there's actually a large study in Germany that's concluded where they are able to increase their predictability scores based on just testing in children year after year and measuring autoantibodies over time. So I, in my laboratory, work in this stage 2 phase, this right in the middle there, where there's still some beta cells left. And the idea is that can we identify drugs that will slow the progression of the disease or even reverse it completely? So as a physiologist, we work largely in rats and mice, and and I won't go through all the details on the slide because that would take too long. We worked with this beta cell toxin called streptozotocin, and what we tried to do is model what we think is a good estimate of what beta cell function and beta cell volume is, or numbers of beta cells in a human, such that we can titrate the dose of this drug and then study therapeutics. So we do this fancy test called a hyperglycemic clamp, where we implant catheters into the carotid artery and into a carotid artery and jugular vein of a mouse. So then we can study them without touching the animal. Stress is very detrimental to metabolic studies, and so everything we do in the animals at this point forward is we don't touch the mouse. So they're sitting in their cage, and what we're looking at is more close to reality. So we measure beta cell function then at the end of this, and what we found is that this one specific dose of the drug, we're able to get in that sweet spot of stage 2 of the disease. So we're looking for molecular pathways and drug targets. We focused on this particular protein called MIG6. It's in the EGF receptor family signaling cascade. And the reason we're focused on EGF receptor signaling, so if anybody's heard, you've probably heard about this in cancer, where it often goes awry. And it turns out there's actual SNPs or genetic variants in specific proteins of this pathway that can be a susceptibility loci for type 1 diabetes. Nevertheless, so we got interested in ways that we could stimulate this pathway. And it has an endogenous brake, and that's this MIG6 little C-shaped thing floating around. And so our idea was, could we knock this out in rodents and somehow reverse or prevent their diabetes? And long story short, that is what we found, although something unexpected came along our way. And this is a very busy slide, so I'm just going to skip it. One thing that we found is there's actually a doubling of the alpha cell mass. And so we looked past this, to be honest. And it wasn't until we got later on that this became important for us. So we did what's called a glucose tolerance test on the bottom left there. And the two top ones are aberrant glucose tolerance. So those are mice with diabetes. When we knock this gene out in the whole pancreas, there is some improvement, so that's great. There is actually an improvement in beta cell mass, so the numbers of beta cells. What I didn't tell you, what was on that first busy slide, was that the numbers of beta cells in these genetic variants or genetic knockout animals is the same as the control litter mates, so the brothers and sisters of those animals. So it turns out when we give this drug, they don't actually lose beta cells. Now, that's not maybe totally correct. What we noticed at the end of the study was that, remember I told you they had twice as many alpha cells. At the end of the study, after the drug, the alpha cells go to normal levels. And the idea is that those alpha cells have actually converted into beta cells. And why is this important? So where do we go from here into humans? So well, it turns out people with type 1 diabetes have plenty of alpha cells. They don't always function as well, but there's plenty of cells. So we're now trying to develop therapeutics that will allow us to target the same system or the same process in humans. Can we convert existing alpha cells into beta cells in humans? And so, what does this have to do with precision medicine? A little bit because this really relies on precision diagnosis and precision testing of pharmaceuticals that are being developed, right? So we would know that the drugs we develop maybe are not going to work for someone who's had longstanding diabetes for 20 years, or maybe has different kind of genetic susceptibility. So our hope is that we can at least identify one subclass of patients that this would work for. And what gives us hope is that when you look at all of the historical trials to prevent or reverse type 1 diabetes, most have been prevention. They've been largely focused on the immune system. There's always a small subclass of, quote, responders, but the therapies never get approved because it's only maybe 8% of the people on it. So you're looking for 100% success rate. That's just not going to happen because these people have different diseases. Everyone with type 1 diabetes may have a different type of disease. So again, we're light years away, but hopefully with research and rats like me, we will get a lot closer to having more precision medicine for people with diabetes in the future. And that's all I have. We'll get to the questions, right? Great.
Well, thanks to all of our panelists. That was a good variety of information, lots to think about. And I really appreciate that you brought up future opportunities in health careers, in research, the data privacy issues, and touched on some other ethical issues. I open it up to questions. I know our time for the panel is up, but I think I'd like to spend about 10 minutes taking audience questions, and I have a couple of my own if we don't get questions from the audience. But I'll open it up.
Don't be shy.
I can get
Don't
a
be
start.
shy.
Oh, go ahead.
Yeah, yeah. No, that's a great question. I'm glad you asked. Can you hear me okay on this one? Okay. Yeah, so the other thing that we did wrong in the past is we blame this disease entirely on the immune system and not it's actually a dual, at least two organ system disease. So the beta cells are not an innocent bystander, right? And the other thing we don't know is how many beta cells you have to start. So we're getting a little bit better at actually being able to provide endotypes and more at the molecular level, like you would do for cancer, say, all right, this person has autoantibodies to this, to that, to the other. And if we can then look at genes from the beta cell side, we might know, how involved is the beta cell in actually triggering this event. So it turns out the beta cells, when they're under stress, start making fusion proteins that look like antigens, so neoantigens, and that may trigger the disease. That doesn't happen in every single person. So as we think about therapeutic strategies targeting the immune system, targeting the beta cells themselves, maybe we're even talking about a cell replacement therapy, we kind of need to have a handle on, what are the molecular or what are the genetic changes that allow us to kind of dial in those things. I think the most promising cure is actually stem cell derived beta-like cells. And we're at a stage where we can go in and use CRISPR technology to change the sequence of the DNA and put in precise things. So you wouldn't want to give somebody beta cells and recreate the same disease. So we are going. I'm optimistic. We will in my lifetime, hopefully, unless I get hit by a bus crossing the street. We will have this in my lifetime, if not my children's lifetime, of being able to say, all right, your type 1 diabetes is this. This is how I should hit you immunologically, and this is how I should hit you on the beta cell side. That's sort of the end game, and I think we'll get there. You're welcome.
So I have two questions. First, how would this be applied to aging population? So older people have variety of diseases. They all interact with each other. And second question, and I'll be done. So the main obstacle for using AI is the privacy issues, because you're supposed to kind of share data. Otherwise, you don't have enough information to actually to train your algorithms.
So I'll just say for genetics, we've often thought about genetics as being mostly for those people with those rare, highly penetrant syndromes. But there's some data from the nurses study that show that genetics can be helpful all throughout someone's entire lifetime. So even people who get sequenced in retirement age, there are usually some nuggets in there that can actually impact their care. And that is about like things like heart disease, things that are long-term health problems. Cancer is generally a disease of older people. So again, if we can do genetic testing to help maybe prevent or successfully treat very early on with cancers, that should help even people throughout their whole life in the sort of syndromes of aging. For the privacy issue, though, it is a concern. I think we do have AI algorithms that can be trained on large population data sets to be the sort of discovery set. And so I personally, as a genetics researcher, am very thankful to those people who volunteered to be in like the UK Biobank and the All of Us data set, because it is a bit of a risk to put your data out there for researchers to use and to be available on the internet from someone who just takes a little course and signs off that you're going to trust that all these people are going to use their data properly. For the actual testing, though, for patients who, and I'll let our fellow panelists talk about it, once we have those workflows sort of optimized, you can keep the data of the individual patients hopefully quite safe by doing that all on a local computer. You don't have to necessarily put it on the cloud. The efficiency of the algorithms is getting better so that they can be run on local computers. You don't need a high-performing computer cluster to do the kind of analysis that would take to interpret someone's genome and then make a clinical decision. And then once you have the interpretive report, you don't necessarily have to save that data. If we had, like, let's say the Kaiser system had whole genome sequencing for all of their people, it would crash their system. Like, there's no way to store that much data. But sequencing is so cheap now, there's really no reason to store it. If you ever need it again, just sequence it again. You will save money over time than if you just keep that data on a computer somewhere. I'll let you talk.
I'll just say it, at City of Hope, every patient who comes in the door signs, if they choose, signs an informed consent to be sequenced, right? It's just part of thing. And the UK Biobank, like, so someone that looks like me is going to benefit from things like the UK Biobank. Is that going to help, you know, people coming from Asia? Maybe not as much, but there's huge data sets. Europe is like light years ahead of us in banking and doing all this. So great, kudos to them. But the privacy, I think we're just going to be in an era where everybody knows everything about you, and you know, oops, you know.
So just to add on that, for genetic data privacy, at least for us, when we transmit it after it's done, like I said earlier, we have the ability to remove it. It's an opt-in, opt-out. Not many companies actually uniformly do that. But at the same time, when it comes to AI and everything, most of the genetic data is already de-identified. So at the most, they're only going to see that you're a male or you're a female, and that's about it. But at the same time, with the growth of AI, I think that it would be helpful to encourage more use of blockchain technology, more end-to-end encryption, because even for us on a daily basis, you do your tests, when we create the report, it's gone at the end of the day. The person who interpreted no longer has access to your data. It's stuck with us. And whatever they used on their computer disappears as well. So I think it's, with anything AI, it's about who you trust. And again, it also comes down to the individual who's accessing your information. that makes it important as we develop more mechanisms for the future. So.
Well, thank you, everyone. Thank you for all your questions this afternoon and for our panelists for joining us, for all your insightful comments and lectures today. So we just wanted to welcome everyone to reception. If you had more questions, I'm hoping our panelists can stay for a few minutes and maybe the rain calm down, but please join us. And thank you all for taking your time today and continue to connect with, of course, the sciences with Fulbright and join us for more symposiums in the future. Have a good afternoon. Thank you.